Hi, I'm Associate Professor Sam Forster. I lead the Microbiota and Systems Biology Laboratory at the Hudson Institute of Medical Research, and I'm also Chief Scientific Officer at BiomeBank. Every one of us carries thousands of bacteria, millions of individual cells, and they're intimately associated with all sorts of sites on our bodies. So our gut is the most common place we think about, but our lungs, our skin, everywhere where there's an interface with the external environment, there's bacteria and other microbes that actually enable us to interface with the external world. They detect changes and they regulate our immune response. And this can be really important from a health perspective because they can tell us that things are going well, we should have a healthy state, or something's gone wrong, we have an inflammatory response, and that can then be detrimental or it can help clear a particular condition. The microbes we carry are intimately associated with our health, and we're now seeing these associations with a whole range of conditions, from inflammatory bowel disease to infections like Clostridium difficile infection, but also conditions like lung disease, autoimmune disease, cancers. And what we're really starting to appreciate is that the right mix of microbes at the right time for the right individual could be a cure, and at least it could be a therapy that reduces the severity of the condition. Each one of us carries a diverse array of microbes, and what we need to do is understand why we carry these microbes, how they're linked to our health state, or why they cause disease. What we know is that through exposure in the environment, through food we eat, through other sources, we're exposed to not only pathogenic or disease-causing microbes, but also the health-associated microbes. We need to understand which microbes we carry, why we're carrying them, and then how we can change that composition such that we can improve our health state. This could involve changing our diet, it could involve adding specific microbes at specific times, or it could involve removing the microbes we have completely and replacing them. What we know is that the microbiome interacts directly with our immune response. It can modulate the immune response through activating and causing disease, or it can suppress the immune response and actually reduce symptoms. If we can understand these interactions, it offers the opportunity to then change the response and cure disease.
My name's Sam Costello. I'm CEO and co-founder of BiomeBank. BiomeBank is a biotechnology company, so we make and develop microbiome therapies. And we're particularly focused on the gut microbiome. So these are the microorganisms that live in the human gut, and they're really critical to our health. So we've outsourced many of the important jobs that need to be done in our gut related to digesting food, maintaining the barrier of the gut, immune regulation, many of these things to the gut microbiome. And so if we lose these organisms, we can lose the important functions they perform for us. And if we lose those functions, we can develop disease. And so there are an increasing number of diseases that have been associated with this problem of loss of gut microbes and loss of function. And thankfully we know now through studies, interventional studies using fecal transplant, so in some cases we can actually treat disease by restoring these missing microbes. We've co-evolved with the microbes that live in our gut, and therefore and we've co-evolved with them over a long period of time when our relationship with the environment was quite different. And so there's been a large number of changes in our environment, particularly over the last 50 years or so, to do with diet, so much more processed food, lower in fibre, as well as a number of hygiene issues where there's a lot less opportunity to transmit organisms between people. And so this has resulted in the loss of gut microbes. And another particular change during that period of time was the development of antibiotics. And that has been a remarkable benefit to humanity, has allowed us to treat many of the most lethal diseases that were afflicting humans prior to the 1950s. But one downside of the overuse of antibiotics, particularly in the food system, has been damage to the gut microbiome and disease developing as a result of that.
At the Hudson Institute, we're focused on fundamental research, understanding the microbes, understanding how they interact with each other, and understanding how they interact with our host cells. Through that understanding, it opens the opportunity to develop new therapies. But we can't do that as a research institute. we have to do that in partnership with industry. And the industry partner that we work most closely with is BiomeBank.
So we're at the new facility on North Terrace. We've just opened the facility with both the federal and state health ministers. And we were here with all of the staff, investors and other people who have helped us in our journey since our founding in 2018.
We have some incredible clinicians, some incredible researchers here in South Australia, but we know that there's so much more that we can do to improve our research and, importantly, the commercialisation and translation of that research into improved clinical care.
BiomeBank develops and delivers microbiome therapies. So we have the world's first approved donor-derived microbiome therapy that was approved in Australia in 2022 for the treatment of C. difficile infection. And we use that therapy in clinical trials to identify organisms that may be therapeutic for other disease indications. And so we're developing cultured therapies for 2 disease indications. So these therapies are human gut microbes that have been grown in the laboratory in a bioreactor, as opposed to collected directly from a human donor.
So this precinct is incredible. So we're here on the western end of North Terrace. We've got the University of Adelaide Medical School, SAMRI 2, that has the Proton Therapy Centre. SAMRI, the Medical Research Institute. And then just across the road, so we've got the Royal Adelaide Hospital. So that interconnectivity with the biomedical precinct will be really advantageous. And we're really, really excited to be a part of this community now.
We work with a range of hospitals and universities around Australia. We have agreements with the Hudson Institute of Medical Research, St Vincent's Hospital in Sydney, St Vincent's Hospital in Melbourne, RMIT University and the Central Adelaide Local Health Network. Australia has world-class medical research and is really in the top 10 globally in terms of output in that space. Where we have perhaps struggled in the past is in the translation of that research into therapeutic products. And so we are really determined to help improve that situation, translate a lot of the fantastic research that is occurring in these institutions to develop therapies here in Australia for global populations.
The Australian Microbiome Culture Collection contains over 40,000 bacterial isolates. This provides a resource that enables people to say, I want a particular species or I want a particular set of species, and use those to do experiments. These experiments can include everything from in vitro bioreactor work, where we're just understanding how species 1 interacts with species 2, through to more complex models where they might be animal models where we introduce particular mixes of species, or cell-based models where we can start to understand sequentially the interactions between particular bacteria and then how nutrients, antibiotics, immune changes modify those communities. All of these opportunities are available because we have this vast collection of microbes where we can say this particular combination for you is relevant for this experiment. Taking clinical samples, we then can replicate the host environment in using microfluidic organ-on-a-chip systems. This allows us to take human cells, combine them with specific communities of microbes, and see what response they induce. In doing this, we can try new therapeutic combinations and see if in your cells, with your genetic background, with your immune priming, do you get the same response, or can you modify that response just by changing the microbiome and the nutrients available? This gives us huge power to start to say, well, if we were to change your microbiome in this particular way, this is the likely immune change that we would achieve.
An example of a collaboration agreement we have is with St Vincent Hospital in Sydney, where we're collaborating with a group of researchers looking at checkpoint inhibitor-induced colitis, and using our donor-derived product to look for a clinical signal, but also to look at the microbiome associations with any improvement seen clinically, with the ultimate aim of developing a cultured therapy for that condition.
The partnership with BiomeBank allows us to translate these fundamental research discoveries into understandings that can then benefit patients. It's important, obviously, to separate these two elements because fundamental research can chase novel or exciting things that don't directly benefit patients but may indirectly provide value. So by focusing the fundamental research in Melbourne, we can understand the key elements of the biology that then feed into the design of therapies that BiomeBank's going to implement for patients. In Adelaide with BiomeBank, we can learn from the understandings, the knowledge, the fundamental experimental capabilities available here at Hudson and use that to optimise and fast-track therapies for patients.
We see microbiome therapies evolving in the next few years from predominantly donor-derived therapies that are harvested from screened healthy stool donors towards cultured therapies that can be grown in the bioreactors and produced under controlled conditions, because cultured therapies would have a number of advantages. We could control the composition, so we wouldn't have the variability that comes from a donor-derived product. And so with that controlled composition, you have more control over the mechanism of action, and also far more easy to scale at potentially lower costs of production as well. And so those cultured therapies can be constructed with specific mechanism of action targeting specific diseases. And so we see that there have been a number of breakthroughs in the field that have allowed these cultured therapies to be possible. And our Chief Scientific Officer, Sam Forster, has been involved in some of those breakthroughs in culturing these previously thought to be unculturable organisms.